Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, in some of the previous lecture, we have already studied about the structure of DNA, its discovery, its properties, and how it is packaged in the form of chromosome. That all thing we have discussed. Now today, we are going to talk about some of the experiment which have been done in the search of genetic material or in the search for genetic material. So, if we talk about this topic, we know that Gregor John Mendel, Walter Sutton, Thomas Hunter Morgan, and other scientists had narrowed the search to the chromosome located in the nucleus of mouse. Because by the Mendelian discovery, Mendel said about the factor, then Thomas and Morgan and others, and Walter Sutton and Bovary given a chromosomal theory of inheritance. So based on this, ultimately the thing was shorted like that. These genes are present on nothing but a chromosome, and this chromosome is located in the nucleus of most cells. Now, later on, this discovery is led to the some of the more conclusions, and in that some of the experiments have been done for the search of genetic material. So first is the transforming principle or experiment we are going to talk, and this transformation experiment is carried out by Griffith. So if we talk about this experiment was first performed by a British bacteriologist, Frederick Griffith, in 1928. In this, he carried out series of experiments with the bacteria. And in a series of experiment, Streptococcus pneumonia or pneumococcus if we talk, which is also known as Diplococcus pneumonia. When we talk about this bacteria, they worked on this bacteria. Either it is known as Streptococcus pneumonia or Pneumococcus pneumonia or Diplococcus pneumonia. And this bacteria is known to cause a pneumonia in mammals, including human. So Griffith studied the two strains of this bacteria, that is Streptococcus pneumonia or Diplococcus pneumonia, or we can also call that as Pneumococcus pneumonia. And he studied that two strains are there, one is S strain and one is RS strain. Now this S strain is considered as virulent or disease causing. Means it is able to cause a disease. Why? Because this S strain of this bacteria possess a mucilase covering. And they give a smooth or regular colony. And they have ability to cause a pneumonia. Whereas if we talk about R strain or a non-virulent strain, then this strain lack a mucilaginous sheath. And due to the lack of mucilaginous sheath, it forms a rough or irregular colony. It forms a rough or irregular colony and it is unable to cause disease. So the important thing is that why they are giving a regular or smooth colonies and why they are able to particularly cause a disease that is a S strain, because they have capsule of glycocalyx. We know that glycocalyx can be of smooth layer, can have a capsule, or they can have a slime layer. If capsule is then they will produce a smooth or particular shaped colonies, and they are able to, because this capsule has a virulence. But this R strain has slime layer, and this slime layer is not giving a perfect sheet, and due to this, because they lack a mucilase, so this mucilase is present in capsule but not in a slime layer, so they forms a rough or irregular colony and they are unable to cause a disease. The series of experiments of Griffith are as follows if we talk. In experiment, he developed this S strain, RN strain, taken this strain, and they have caused this infected to the mice. So in this first, they have injected S strain, which is a virulent strain, into the mice and developed a pneumonia. And at the result of this, at the end, the particular mice has died. So the living S strain, if we inject it into the mice will die and due to the pneumonia. And from the cells of this pneumonia, the particular living S cells are recovered. But in another experiment, another setup, he injected R strand, which is a non-virulent, into the mice, and due to this non-virulence nature, it is not able to cause a disease, and mice survived. So living R strain recovered in the body of mice. So when R strain injected, the mice is alive and survived because R strain is non-virulent. But in the third setup, he heat killed S strain bacteria and then injected into the mice. And here, when they heat kill the S strain, which is a virulent strain, but after heat killing, they injected into the mice, and mice did not get the disease and remained alive. So when he injected heat killed S strain into the mice, mice survived. Okay, and the S or R strain cells are not found in the body of mice. Now the question is how this heat killing is performed. Simple. You can take a particular S3 type. Actually, S3 type is taken and R2 type is taken, and they have heat kill like this. Okay, so this S3 type bacteria are going to be known as heat killed. And then injected heat killed S strain, which heat killed S strain is not able to cause disease, and R strain is also not going to cause disease because both are non-viral case. But when both are injected into the mice at a same time, it is seen that the mice particularly died. So heat kill S strain, if we injected along with the R strain into the mice, then the mice died due to the pneumonia. So here be remember it is heat killed S and plus R strain. And then Griffith concluded that R strain bacteria had somehow been transformed by the heat killed S strain. So Griffith concluded that might be both R strain is non-virulent and heat killing state S strain also become non-virulent because in that mass death does not occur. So if we give combinedly, then the death is occurring. Why? By thinking so much, Griffith concluded that might be R strain bacteria had somehow developed or transformed by the heat kill S strain bacteria into the virulent bacteria. Means R strain become virulent from the non-virulent state. How? So some transforming principle, or we can say transforming chemical, actually transferred from a heat killed S strain to the R strain, and that enable R strain to synthesize a smooth polysaccharide coat, and that enable the R strain to synthesize a smooth polysaccharide coat and become virulent. Okay, so it is showing that heat kill some material or some transforming chemical from the heat kill S3 strain received or absorbed by the R strain, and R strain become virulent because they develop a capsule. So this must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from the experiments of the Griffith, that which chemical has transported or which chemical has moved from the heat kill S3 to R2, that was not particularly given or concluded by this experiment. But later on, biochemical characterization of this transforming chemical or transforming principle was done. And prior to the work of Oswald Avery, Colin MacLeod, and Maclean McCarthy, the genetic material was thought to be a protein. Initially, it was thought that the genetic material is going to be a protein. But Oswald Avery, Colin MacLeod, and Maclean McCarthy, if we talk, this are particularly before them, it was thought that it is going to be a protein. But They performed an experiment, and these scientists worked to determine the biochemical nature of transforming principle in Griffith experiment or transforming chemical. Now, what they did? They purified biochemicals, that is a protein, DNA, RNA, etc. from the heat-killed S cells. From the heat-killed S cells, they have isolated this all protein, DNA, RNA, etc. to see which one could transform live R cells into the S cells. So for that, they done an experiment and they discovered that DNA alone from S bacteria caused R bacteria to become a transformed into the virulent form. They also discovered that protein digesting enzyme proteases and RNA digesting enzyme RNases did not affect transformation. So the transforming substance suggesting that the DNA caused the transformation. And they concluded that DNA is the head material, but not all biologists were convinced. So here the thing is that what he did from S3 bacteria, they have separated proteins, RNA, DNA, and they inserted these proteins from the S3 into the test tube which having a R2 bacteria. This having a R2 bacteria. In A tube they have kept this one. In B tube they have taken R2 bacteria and they have injected RNA. In this also they have taken R2 bacteria and they have injected DNA. Now in these three bacteria they have taken R2 and injected DNA. The R2 remained R2 in this strand. In this also the R2 remained R2. But in this, because R2 remained R2 means non-virulent. But here R2 become virulent when they injected into the mice. So means R2 become S3 or R2 become virulent, we can say. But scientists was not confirmed yet. So what they did, in this they have inserted DNA and along with they have inserted DNAs in this tube with the R2 bacteria. Now this DNA is cut DNA, then there should not transformation occur, and that happened. The R2 remained non-virulent, means they have not transformed when DNA is added along with the DNA. Means DNA is cleaved this DNA. So this way, this particularly from this experiment, there was confirmed that the biochemical nature of transforming principle or transforming chemical is nothing but a DNA. Okay, so this is the experiment which was done by Avery, MacLeod, and McCarthy. So the genetic material is DNA was not completely proved, but biochemical nature of transforming chemical was proved by the Avery, MacLeod, and McCarthy. In continuation of that, to search the genetic material, which is a DNA, Hershey and Chase experiment, after Griffith experiment, Hershey and Chase experiment was carried out. It is known that this is the unequivocal proof. It is an unequivocal proof that DNA is the genetic material. It is an unequivocal proof that DNA is a genetic material. Came from the experiment of Alfred Hershey and Martha Chase. Who carried out this experiment? Alfred Hershey and Martha Chase. This is unequivocal proof, means it is a perfect proof which is given by Alfred Hershey and Martha Chase for the DNA as a genetic material. This is the pair of them. Now in this they used a bacteriophage. And they worked with viruses that infect bacteria, that is bacteriophage. And the bacteriophage attaches to the bacterium and genetic material then enters the bacterial cells. Means the DNA of the bacteriophage or bacterial viruses, which is known as genetic material, enters the bacterial cell by the infection. Then the bacterial cell treat the viral genetic material as if it was its own and subsequently manufacture more virus particles. Means what happens after entering the DNA or bacteriophage, it take over the machinery of the particular, we can say, bacteria, and carry out a lytic cycle where it synthesizes protein machinery, capsid, as well as replicates its own DNA, and then packaging will occur, and then there is a breakdown of the cell wall occur, and the new bacteriophages are released by the lysis of this bacteria. So, such a way more virus particles are prepared. They tried to observe with component of bacteriophage, that is either protein or DNA entered bacterial cell. They tried to observe this component of bacteriophage, either protein is entered or DNA entered bacterial cell, and direct reproduction of the viruses they have observed. Hershey and Chase grew viruses. Now, what the in experiment it was did? They grew viruses on a medium that contained radioactive phosphorus, and some others viruses on the medium that contained a radioactive sulfur. So when we particularly culture in such a petri plate, if I keep one petri plate with the medium of P32 and another petri plate with the medium of S35, and if we culture the viruses, then the viruses grow in the presence of radioactive phosphorus. P32 contain radioactive DNA because this phosphorus is present in DNA. So DNA will enter into the during a replication. DNA will enter into the DNA. Phosphorus will enter into the DNA, and this way viruses grow in the presence of radioactive phosphorus in a culture medium, and they obtain this radioactive phosphorus in their DNA. So DNA, because DNA contains phosphorus but protein does not. So this happens. So if we talk, this is the bacteriophage structure, and here phosphorus get incorporated into DNA when it is grown in culture medium. Same when it is grown in a sulfur containing medium, it will encounter sulfur, but sulfur is not present in DNA. It is present in which one? Amino acid, sulfur containing amino acids. So capsid will be labeled by the phosphorus. So radioactive phages were allowed to attack bacteria. Now we remember. Now there is a phase. We remember this S35 is become part of capsid as capsid is proteinaceous. As capsid is proteinaceous. Now radioactive phages were allowed to attach to E. coli bacteria. Okay. Okay. So S35 become part of this capsid. So we have a one. Suppose we have two bacteriophages. One bacteriophage with the DNA labeled with the P32, and another bacteriophage with the protein capsid labeled with the S35 isotope. Now radioactive phages, these two which are prepared like this, were allowed to attack to E. coli bacteria, and when it attack, it insert its DNA into the particularly bacteria. Then, as the infection proceeds, the viral coat were removed from the bacteria by agitating them in the bladder, and such a way the virus particles were separated from the bacteria by spinning them in a centrifuge. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a bladder, and the particular experiment will be carried out. So, if we talk about this experiment in more detail, then you will come to know that it is occurring in three phases. Okay. This particular experiment is carried out in three phases. So, let's talk about the three phases which are included here. So, three steps of experiment. Number one is infection. Number two is blending. And number three is centrifugation. So here what happens in infection, the bacteria are taken. These both bacteria have their own bacterial DNA, and in first they are particularly infected with the bacteriophage with the S35 labeled protein capsid. When S35 labeled protein capsid is there, and that particularly inject this DNA. But in another part, the P32 labeled DNA containing bacteriophage attack this bacteria. So when they attack, they insert their DNA within them. After this whole process is carried out within the test tube. After this, blending is done where the test tube in which the bacteria and bacteriophage is infecting the bacteria is agitated in a blender to break the contact between virus and bacteria. So here now this is a particular bacteria with its bacterial DNA and viral DNA, and protein getting separated. But here it has bacterial DNA and viral DNA, and this way protein coat is separated. Now we know that the protein coat here is S35 labeled and here DNA is P32 labeled. So blending is done, agitated so they get separated, and later on centrifugation is done. And when centrifugation is done in the test tube, it is known that here the cell will settle down in this part, and this protein coat remain in the upper part. So here in centrifugation is done, then here you will see the cells and here you will see the supernatant. So here radioactivity detected in supernatant. In supernatant. And here no radioactivity. So obvious why radioactivity is on the supernatant, because the protein this bacteria are going to be dense or they are more, larger in size. So bacteria settled at the lower position and they have not shown radioactivity. But here protein coat has the radioactivity, means sulfur is incorporated in the protein and it is not entering within the cells. But here the cells are there and supernatant is there. Now here no radioactivity in supernatant. No radioactivity in supernatant, but radioactivity detected in cells. And when radioactivity detected in cells, obviously here a DNA was P32 and phosphorus radioactivity is in here, means viral DNA has entered within the particular cells, means this proves that DNA is entering within the cells. So after centrifugation, the bacterial cells showed the presence of radioactive DNA labeled with the P32, while radioactive protein labeled with S35 appeared outside the bacterial cell in the medium. So labeled DNA was also found in the next generation of the phase. Bacteria that were infected with viruses that had radioactive protein were not radioactive. So this indicated that protein did not enter the bacteria from the viruses, but DNA is therefore a genetic material that is passed from viruses to the bacteria. So this proved ultimately that DNA is a genetic material. So bacteria which were infected with viruses that had a radioactive DNA were radioactive. Bacteria with their infected with the virus that had a radioactive DNA means P32 labeled. This bacteria is going to be radioactive and indicating that DNA was the genetic material that passed from the virus to the bacteria. Bacteria that were infected with the viruses that had a radioactive protein were non-radioactive. This bacteria was not does not show any radioactivity. Means that protein is not entered within the cell. So this indicated that protein did not enter within the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. So let us discuss some of the simple questions that transformation experiment was performed by. Obviously, we know that it is obviously done by Frederick Griffith on the mice. Pneumonia is caused by obviously Streptococcus pneumoniae. And third is a genetic material is nothing but it is a DNA. Okay, so this is the two experiment and ultimately the unequivocal proof given by Hershey and Chase. Now after this we are going to discuss the properties of this genetic material, which should be the genetic material that can be described by these properties. So in this, from the foregoing discussion, it is clear that the debate between a protein versus DNA as the genetic material was unequivocally resolved, unconditionally resolved by the Hershey and Chase experiment. It became an established fact that it is the DNA that acts as a genetic material. Now it is clear that DNA is nothing but it is only acting as a genetic material. However, it subsequently became clear that in some viruses, RNA is also acting as genetic material. In some viruses, you will see the presence of RNA as well, like HIV. So, like tobacco-mosaic virus, QB bacteria virus, HIV, all have RNA as genetic material. And bacteriophage, tobacco-mosaic virus, UV, RNA core is there, central core is formed, and they all have RNA as genetic material. So, a molecule that can act as a genetic material. So, it is not only that DNA will always act as genetic material in all organisms. It is not so. RNA can also be there, genetic material in some of them. But DNA is more common, yes. So a molecule that acts as a genetic material must fulfill the following criteria. So whatever the genetic material, it should fulfill the following criteria. And first, it should be able to generate its replica. Means it has a capacity of replication. It should be able to make its own copy. It should be chemically and structurally stable. Chemically and structurally be stable. It cannot be broken up easily. It cannot be mutated easily. It cannot be particularly damaged or broken up easily. And it should provide a scope for slow changes, slow mutation that are required for evolution. Yes, sudden large mutations are not needed, but slow mutations to some changes they can allow, which is necessary for the evolution. Then it will be act as a genetic material. And it should be able to express itself in the form of Mendelian character. Means it should pass from generation to generation in the form of Mendelian character and show its characteristics. Because of the rule of base pairing and complementarity, both the nucleic acid DNA and RNA have the ability to direct their duplication. They can direct their duplication because they have a complementarity law. The other molecule in the living system, such as protein, failed to fulfill the first criteria. It is not able to generate its copy by its own. So it cannot exert the genetic material. Whereas if we talk about RNA in comparison to DNA, RNA is not predominant and very suitable genetic material because RNA is highly reactive and unstable. Why it is highly reactive and unstable? Because it has a 2-OH group. On its pentose sugar, you will see the presence of OH group in ribose sugar. In every nucleotide in RNA, and it is a reactive group, so it makes it labile or easily degradable. Any molecule can react with this easily. RNA is also now known to be catalytic. It has some catalytic role also. It can speed up some reaction as well. So it functions as an enzyme, therefore reactive and unstable. And uracil is present in RNA, which is less stable as compared to the thymine. That is methyl uracil of DNA. If we talk about RNA, RNA being unstable mutated at a faster rate, and consequently the virus having a RNA genome and having a shorter lifespan mutate and evolve faster. So there is a more evolution in case of the viruses. So due to this, it is somewhat looking that DNA is going to be the genetic material, and DNA is the predominant genetic material of organism. It is chemically less reactive. Why it is DNA is selected as genetic material? Because DNA is chemically less reactive, it is structurally more stable compared to the RNA. And DNA stability can be clearly evident in Griffith transforming principle. It is clearly evident in Griffith's transforming principle that DNA remains stable. Heat which kills the bacteria at least did not destroy some of the properties of genetic material. See, one of the important, it can destroy everything, but it cannot destroy all the properties of genetic material or DNA, specifically DNA. This now can easily be explained in the light of DNA. So the two strands of the DNA being complementarity, if separated by heating, they come together when the appropriate conditions are provided. And presence of thymine in DNA instead of uracil provide additional stability to the DNA. So due to this thing, it is more stable. The hydrogen between purine pyridine and their stacking one over the one base pair over the another base pair make DNA more stable for strong genetic information. And here DNA is capable of undergoing slow mutation required for genetic material. So mutation are possible, slow mutation will occur, but it cannot easily degradable, it is stable. And one more, it has a power of repairing also. So if mutation can occur, they can be recovered, or if any problem occur in DNA, that can be repaired. So both DNA and RNA involved in a genetic expression, because RNA can directly code for the synthesis of protein, because from DNA the information passes to the RNA and the RNA forms the protein. And so RNA can easily express the characters if we compare DNA and RNA, because DNA will undergo transcription and form RNA, and RNA will undergo translation and will form a protein. So DNA is dependent on RNA for synthesis of protein, and the protein synthesizing machinery evolve around RNA. But DNA being more stable is preferred for a storage of genetic information, whereas RNA is better for transmission of genetic information. So if we need to talk about the transmission of genetic information, then RNA is better. But if we want to talk about storage of genetic information, then DNA is better. So this is the comparison actually between the DNA and RNA. And by this, it ultimately proves that DNA is going to be the genetic material. So based on which experiment DNA acts as a genetic material? So Hershey and Chase experiment given an unequivocal proof that suggested that DNA is a genetic material. Which base pair is present in RNA instead of thymine? Instead of thymine in RNA, you will see the presence of uracil. DNA is dependent on blank for synthesis of protein. DNA is dependent on RNA for the synthesis of protein. Okay, dear students. So in this lecture we have discussed about the some experiment in search of genetic material and why DNA is acting as genetic material. That also we have discussed. Okay, so I hope you understood. Thank you.